Translation occurs at ribosomes and has steps: initiation, elongation, and termination. Messenger RNA, ribosomal RNA, transfer RNA work together to join amino acids into polypeptides and proteins, and retroviruses kind of alternate the flow of information. So as we talk about this flow of information, we go from DNA to RNA to protein or polypeptide. And again, transcription goes here, translation goes here. Now retroviruses, because their genetic material is RNA, when they have to use the host cell in order to get back and reverse that flow of information. So instead of going from DNA to RNA, which it eventually will, it needs to go from RNA to DNA. So reverse transcription happens with reverse transcriptase. And that takes the viral RNA, turns it into DNA, which gets embedded into the host DNA, which then can follow the normal flow of information of transcription and translation. So as we focus on translation here, we're talking about ribosomes. And ribosomes have small and large subunits. The only time they come together is when they're actually doing translation. Otherwise, they kind of exist apart from each other. And as translation happens, it doesn't just happen one ribosome at a time. The messenger RNA will be fed into sequential ribosomes, so many proteins can be made all at the same time. Prokaryotes have free-floating ribosomes, and so do eukaryotes, but eukaryotes will also have ribosomes attached to the rough ER. Both of these organisms will have thousands of ribosomes. So eukaryote, again, because there is a nucleus, you can have some processing of the messenger RNA, but then these processes of transcription and translation are going to happen at different times because the messenger RNA has to leave the nucleus to get to a ribosome. However, with prokaryotes, transcription and translation happen simultaneously because as RNA polymerase is making the messenger RNA, it's feeding directly into ribosomes, so both of these processes are happening at the exact same time. Transfer RNA has this unique shape, and it is the codon from the messenger RNA that hooks up with the anticodon on the transfer RNA. These extra wings help it bind with the large ribosomal subunit, but every codon and anticodon pairs with a specific amino acid, and we use this chart to help understand that. So with this codon, it's CUU. So we go C for the first base, U for the second base, and U for the third base, which coordinates with the amino acid leucine. When you see this chart, we have a start codon. The universal start codon is AUG, which codes with methionine. And methionine doesn't only exist at the start, it can exist further along the polypeptide chain. The other thing to pay attention to is that there are 3 stop codons, which will cause termination to occur. So we start with initiation, and the small ribosomal subunit binds with the messenger RNA. And at that messenger RNA, we have the binding of the first transfer RNA from the start codon AUG. So methionine is going to kick things off. From there, the large ribosomal subunit hooks on, and it stabilizes the transfer RNA. And there's 3 sites. We have the E site, which is the exit site, the P site, which is the peptide site, and the A site, which is the amino acid site. Elongation is basically what it sounds like. We're going to assemble the polypeptide chain, one amino acid at a time. So 2 transfer RNAs can hook on to the ribosome at any given time. So we have the peptide in the peptide spot, and we have the new transfer RNA landing in the amino acid spot. Now, the ribosome, through enzymes, or ribozymes, are going to help form the peptide bonds that join these covalent peptide bonds that join the amino acids together. And now this is going to shift. This transfer RNA that no longer has the amino acid connected to it will get pushed to the exit site and it will get released. And that peptide will be at the peptide spot, and then the new amino acid transfer RNA can land. Eventually, we're going to get to termination, and a protein release factor will bind to the stop codon. And when this gets to the P site, it disconnects and releases the polypeptide chain to coil up into its two and three-dimensional shape to become a functional protein. And then the large and small subunits disconnect and dissociate. So we have our transcription happening. The messenger RNA is going to the ribosome, which has large and small subunits. At the ribosome, the transfer RNA will bind, and the ribosomal RNA will help form the peptide bonds until it hits the termination sequence, where the protein will be released, the messenger RNA will be released, and the subunits will dissociate. So again, translation occurs at ribosomes through initiation, elongation, and termination. Messenger RNA, ribosomal RNA, transfer RNA combine amino acids into polypeptides and proteins, and retroviruses kind of alternate flow of information.